And with that, it is my absolute pleasure to introduce our speaker today. If you go to the next slide, please. Thank you. So I'm going to be introducing Dr. Carolyn Yrigollen, who is a researcher at the Children's Hospital of Philadelphia with 10 years of experience working on gene editing systems to treat heritable neurological disorders. Carolyn earned her Ph.D. in genetics from the University of California, Davis, where she studied the structure and function of the Fragile X gene. She's currently working on CRISPR-based approaches to correct the underlying mutations that lead to Fragile X syndrome, as well as Fragile X-associated tremor and Ataxia syndrome. And with that, I will turn the floor over to Dr. Yrigollen.
Thank you, Celeste. Yeah, I'm really happy to be here today and talk to you about one of the kind of favorite topics of mine to think about, work on, and that is how we take what we know about genetics, what we've learned, and the tools that we've developed to try to treat neurological disorders. So before we get into discussing gene therapies, I think it's important, as it is for us as we're designing gene therapies, to think about what exactly we want those therapies to do. And so at the very foundation of this is what is a genetic disorder caused by, and what are the early impacts of that genetic disorder. So this slide might remind you a lot of your high school biology class, and when you learned about how all humans and other organisms are made up of a large quantity of cells that are very diverse in nature. We have blood cells, we have skin cells, neurons in our brains, immune cells, and every single one of these cells has its own copy of the human genome in it. And if you also remember from your high school biology class, humans inherit 2 copies of every chromosome, so 23 chromosomes in total, one from their mother and one from their father, and this makes up about 6 billion base pairs of DNA. However, cells are really good at handling all that DNA. So what they do is they organize it into these structures called nucleuses. They pack it up really tightly, but are still able to have certain areas be accessible so that they can express important information. For example, neurons in the brain would make accessible genes that are important for brain function, whereas blood cells would make accessible genes that are important for blood, such as hemoglobin. And the way that they express these important genes is through a process of transcription, where this enzyme called an RNA polymerase will read through the double-stranded DNA, and it'll create an entirely new molecule called RNA. And this RNA is similar to DNA, except it's single-stranded. At the point where the RNA is made, it can then be shuttled outside of the nucleus into the main body of the cell, where it can be either kept for when the cell will need to produce whatever protein it encodes, or it can immediately go on to express a protein. And it does that through a second process called translation. And in translation, a ribosome is going to read through that RNA sequence, and for every 3 nucleotides of the RNA, it's going to add a new amino acid. And so proteins are really just long chains of amino acids that then end up folding over on themselves. Sometimes they add new chemical modifications to them, and then they're able to perform a certain function. When we think about mutations, I think it's important to consider what mutations actually are. In the grand scheme of things, they're very small. So I have put in this zoomed in view of a cell mutation, which if you have good eyes, you'll be able to see this kind of small white blip in the long strand of blue DNA here. We can zoom in a little bit closer and you can see this is where the mutation is. And I did this because I want you to think about mutations and just how big they are compared to that 6 billion bases of DNA that's in this nucleus. They're pretty small, and so when we are trying to target them for gene therapy, we have to bear that in mind. These mutations can be as small as a single base pair, which is depicted here as this single line. on this DNA strand. And then when that gets transcribed into RNA, you can see the RNA has retained that mutation in it. This mutation might be one that would then result in an entirely different amino acid being incorporated into the protein. And that can have really major implications for how that protein functions, and maybe it no longer does function. Mutations can also be larger than a single base pair. You can have large deletions or insertions of entire genes or parts of genes. In ataxias, it's very common to have long stretches of repetitive sequence as the underlying mutation. Or you can have, as I said before, the very small single nucleotide mutations. So long repeats are one of the common causes of ataxia disorders. And you can see this table. You don't have to get a lot of information from here, but I just wanted you to appreciate how many different repetitive mutations give rise to various ataxia disorders. A lot of CAG repeats are causing SCAs. My gene that I work on, the FMR1 gene, is caused by a CGG trinucleotide repeat. And you also have GAA repeats. You have repeats longer than trinucleotide repeats. And these repetitive sequences are unique in that, unlike mutations that are sometimes like a single base pair and often cause dysfunction of the protein, so it no longer functions like it normally would, and we call that a loss of function mutation. Nucleotide repeat expansions cannot only cause this loss of function, but they very often cause a gain of function as well. And this gain of function can be through there being a very long stretch of repetitive sequence at the RNA level. And this causes the RNA to fold up in weird ways and causes it to bind to proteins that it normally wouldn't bind to. Or it could be the long stretches of amino acids that are the same from the repetitive sequence. And this can also ball up in weird ways and make what we call aggregates, which will then bind to other proteins and RNAs. And when it binds to these other proteins, this isn't what normally happens in the cell. And so it ends up binding proteins that have important functions in the cell, but aren't able to carry them out anymore because they're tethered to these repetitive RNA or polyprotein or polypeptide sequences. On the other hand, oftentimes when you have the smaller mutations or the mutations we often think of with genetic disorders, like deletions or missense mutations, so these can be a single base pair that results in a change in that amino acid sequence, or a nonsense mutation, which can be a single base pair that results in the amino acids no longer being produced. So it creates a truncated protein, which is usually not functional. All of these mutations tend to, not all the time, but they tend to lead to more of that loss of function where they impair the protein function, but they don't create a gain of function toxicity. So now that we kind of know what we would want to target with our gene therapies, we can start thinking about how we want to design our gene therapy. The first thing that I like to think about is, and it's an important consideration, where does that gene therapy need to go? So delivering gene therapy is in and of itself a very important aspect to consider. For neurological disorders especially, I would say that there are three kind of top level gene therapy delivery approaches that most people would take. The first is using viral vectors. The second is using just synthetic DNA that you can inject directly at the site you want it to be taken up. And the third is using kind of a mixture of both of those. So the synthetic DNA can be packaged in a particle, but that particle is not viral vector. It's made up of something else, such as a lipid. So with viral vectors, one of the common ones that are used, especially with neurological disorders, are adeno-associated viruses. These are viruses that long ago were developed to be beneficial for biological research and now for gene therapy, because what they did was they took this AAV that normally has a DNA sequence in it that would have the AAV make more AAV, and researchers pulled out that DNA and they put in the DNA that they wanted to enter into cells. And so in order to do that, you still have to retain these two little hairpins structures of DNA. So this is a sequence that will kind of fold in on itself and create this hairpin. But then you can put in any sequence you want in between those. And so this light purple bar here would denote kind of your favorite gene that you were trying to put into a cell. And the dark purple are additional instructions that you're adding with that gene so that the cell will know when to read it, when to express the gene. And that includes this little arrow, which is we call a promoter, and also additional sequences which might be important in stabilizing the RNA, for example. The good thing about AAVs is that they're able to transduce or get into non-dividing cells. So that's important if you're trying to get into, for example, mature neurons. And once they get into those cells, they can establish themselves and be stable in the nucleus and stay there indefinitely so that you can continually express whatever sequence you have packaged into that viral vector. Additionally, current research is being done to make small changes along this capsid that can alter where this vector goes. So it can alter if it goes into neurons or if it would be better going into, for example, liver cells. So you can just change one piece of the protein and it can dramatically change where that vector is going. One of the disadvantages of using AAVs is its small capacity. So it's only able to fit about 4,800 base pairs of DNA. And for example, one of the common gene therapy enzymes that we often want to deliver, CRISPR-Cas9, is 4,100 base pairs. And as I said, beyond the gene that you need to deliver, you also need to have room for these hairpin sequences and for the instructions that go with that gene. So in the lab that I work in, Beverly Davidson's lab, they recently put out a paper in bioRxiv where they are working on developing better improved capsids of AAV that will target more efficiently into the brain. And the way they did this was they designed a lot of different capsids and screened them by injecting them into non-human primates. Why are they injecting them into non-human primates? Because what we find is that between species, AAVs can infect certain cells better than others. And so what we want is to be able to know that when this goes from our work at the bench to clinically at the bedside, that the capsids will have a high probability of going where we expect them to go because non-human primates are so similar evolutionarily to humans versus, for example, mice. And then after they've injected these, the Davidson lab sequenced to see what capsids went into different structures of the brain. So these 2 and 3 letter abbreviations here are different regions of the brain: the caudate, the putamen, the cortex. And then this heat map shows the darker the color, the more capsids got into that region for a specific capsid. So this y-axis is the top 10 capsids for this particular screen. And the lighter the color, the fewer capsids got in. And so doing this, you can then identify capsids that do a really good job going into all of these different brain regions. And so this would allow for broad delivery of our gene therapy. When we then injected that capsid, that top capsid, with a sequence that would create a fluorescent protein, we can see exactly where in the brain the capsid was taken up and was expressing our cargo sequence. And you can see here that there's really robust expression throughout the brain, the various regions, striatum, cortex, the thalamus. Another approach is using the non-encapsulated oligonucleotides. So this can be ASOs, it can also be siRNAs. This is where you deliver either synthetic DNA or RNA directly into the region of interest, resuspended in something like a saline. The nice thing about these are that you can synthesize them with chemical modifications to them that can help with their stability or also influence where they're targeting. But an important thing to think about is that toxicity has been shown to occur with certain ASOs and where they're delivered. For example, it's been shown that some AAVs will show neurotoxicity when delivered into the CNS. And the thought is that this could be because of the amount of negative charge that is associated with DNA and RNA. And it's kind of creating this toxicity. So you can buffer that potentially by resuspending it in a solution that has more bivalent cations. So more positive charges to kind of neutralize that negative charge. And what this recent paper that was deposited in bioRxiv showed was that what they did was they used regular buffer, the buffer that's normally used, and then they made a buffer that has these calcium and magnesium bivalent cations to them, and that's this ACSF. And they show that if the mice are delivered with either of these buffers, but they don't have the siRNA incorporated with them, then the mice are fine. They don't show any toxicity, which here is rated by the amount of seizures or the amount of mice that got seizures following this experiment. But if they add in the siRNAs, they see that the mice where the siRNAs were delivered with PBS are now all getting seizures following the experiment. Whereas the mice where the siRNA is delivered with their kind of bespoke buffer now are not receiving are not getting these seizures following the experiment. So these kinds of experiments are being done to help reduce that toxicity so that this can be a more beneficial approach for treating neurological disorders. One of the scientists in our lab who's working on ASOs shared with me this slide, so you can see the biodistribution in this brain. So you can see here, this is a zoomed in view of a mouse hippocampus, and all of the green signal here is where the ASO is. The blue signal is just so that you can kind of see the architecture of the brain. And you see that there is broad distribution of this ASO 24 hours after delivery. So the third method that we'll talk about is where you would encapsulate your intended cargo into a nanoparticle. And a common one used are lipid nanoparticles. So the good thing about lipid nanoparticles is similar to if you put a drop of oil into a vat of water, it will kind of just create a bead and form kind of a solitary structure in and of itself. The same thing happens with these lipid nanoparticles. So if you put lipids into a solution with your intended DNA or RNA, and sometimes people even deliver protein using lipid nanoparticles, then the lipids will kind of form this shell around the various oligonucleotides or proteins. And this shell can then be used to deliver the cargo into the intended cell. Similar to the ASOs, you can put chemical modifications on these lipids. And this is an extreme example here, but you can see all the different modifications that you can create. So you can put chemical modifications to stabilize it and make the shell hardier, or put chemical modifications on so that the shell releases the cargo more quickly. You can add antibodies that will let it target certain proteins, or other various signatures that would help either in the experiment so that you can track where it's going, or other features. So this paper in biochemistry shows how these lipid nanoparticles were used to create an edit in a DNA sequence. So in this case, the researchers used this very special mouse that was designed to have this synthetic gene put into it. So this gene is made so that it can express this protein called TD tomato. TD tomato is a very bright red protein. It's fluorescent. You can see it. If you look at it under the microscope, you can actually see it from the naked eye if the mouse is expressing enough of it. However, upstream of it are instructions that say, don't express this gene. So these 3 stop codons say, don't express this gene. And so the mouse normally isn't creating TD tomato. But if you have a method using a gene therapy to remove these stop codons, then the gene no longer has those instructions and the cells start making TD tomato. So that would be this TD tomato on version. You can remove these genes in multiple ways. And what they did was they tried 2 different ways. One was this Cre recombinase. So this is a protein that will specifically remove anything between these 2 sites, what we call LOXP sites. They're just sequences of DNA, and Cre is really good at removing anything between those 2 sites. The other method is Cas9, which we'll talk about a little bit later. It's a common gene editor, and it will also target a specific sequence to create a deletion. And so in both cases, you're deleting these stop codons, and you can see from the image on the right that they work at different levels of efficiency. So with the Cre mice, when they inject Cre using these lipid nanoparticles, you can see many cells are expressing this TD tomato protein. Where with Cas9, you do see some cells that are expressing it, and they're expressing it very brightly, but not the same number of cells as when the mice were injected with Cre. All right, so now we know the tools that are available for us to deliver our gene therapy, and we can start talking about the different strategies that exist for gene therapy. So there's a long list of them, and we will give you some examples from each of these that I think are kind of the best hits. So the first strategy is gene replacement, and it makes a lot of sense. If you have a mutation in your gene that is preventing a protein from being expressed or from functioning properly, then if you just replace it with a gene that doesn't have that mutation, it should make the protein that you need and the cell should be healthy. This is a really good strategy to use if your mutation isn't causing that gain of function that we talked about earlier. That's common with a lot of trinucleotide repeat disorders. Because it's not actually removing the mutant gene. So if you're creating a product from that mutated gene that's harmful to the cells, you would still be creating it. You would just also be creating a functional protein. This is also a good approach to use if the protein that you're trying to deliver is small, especially if you're using an approach such as AAVs that has that minimal size capacity that you can use. So the first example that I have of this is from Friedreich's ataxia. In this case, these scientists wanted to develop a gene therapy specifically to target and replace the frataxin gene in a mouse model because the presence of this mutation not only leads to the ataxic phenotypes, but also to the cardiac pathology, and that is what's often lethal. And so the approach they took was to just package the frataxin gene into an AAV. They chose AAV8. So AAV8's capsid targets the heart very well, the cardiomyocyte cells in the heart. And beyond the gene, they also included instructions, including the promoter, the PGK promoter, on when the gene should be expressed. To test whether their AAV would end up being beneficial in making the frataxin protein, they first introduced it into human cells. So here you have 2 wells of cells. In one case, the cells did not get the AAV with the gene therapy, and then in the right panel here, you can see that it did get that gene therapy. The cells were then stained for the frataxin protein. So you can see anywhere where you see green in either of these 2 panels is where frataxin is present, the protein specifically. And so you can see only in the cells that got the gene therapy are they making frataxin. So next, they wanted to look at this in an animal model. So they injected mice that do not produce frataxin, so they have a mutation in their frataxin gene. And that is this second panel here. So these are mice that have a frataxin mutation. And in this case, these are images of hearts, zoomed out and a zoomed in image. And frataxin here is stained in brown. And so this mutant animal, you can see, has no brown. It's all blue staining here, versus the animals that don't have that mutation, the wild type animals, you can see a general brown hue over this entire heart. And then the mice that they injected with their therapy, they either included the frataxin gene with the human sequence or the mouse version of the frataxin gene. And they see frataxin protein expression in both of these cases. They also did a lower dose of frataxin. And you can see here, compared to the higher dose, you do see less frataxin here, but you still do see quite a bit of it. So then what would this mean in terms of survivability of these animals? To test that, they injected animals and then they just let them go until they had to die. And so the survival curve here, you can see the gray bar is the animals that have the mutation and were not given the gene therapy. And these animals only survive about 8 to 10 weeks. Whereas the animals that don't have the mutation, the wild type animals, are this blue line, and they go till the end of when they were tracking these animals. Most of the animals that received the therapy and had the mutation also survived through the length of the study. You can see here that maybe 1 or 2 mice ended up dying at this 17-week time point. But this is really good because this is showing that with this gene therapy, that they were able to extend the life of these animals by about at least twice as long. So this work has been published also by other scientists, and it actually is currently in a phase 1/2 clinical trial through Lexeo Therapeutics. So this is exciting news for Friedreich's ataxia research. Another approach you can take is enhancing your gene expression. So you can drive more of the protein to be produced either by targeting and stabilizing the RNA that's already made, or by recruiting factors to the gene that will encourage more expression of that gene. And so now you're creating more RNA than you normally would. And this is a good approach if you have either a gene that is being expressed at low levels, but if you increase the expression of it, you expect that that will help with cell survival. The other approach is if you have 2 copies of the gene and only 1 copy has the mutation, and so potentially the other copy you can increase expression and have a favorable phenotype. So while I don't have an ataxia example to give for this, I do like this example, which is of a family of disorders caused by mutations on chromosome 15. So this is Angelman syndrome and Prader-Willi syndrome. The unique thing about this is on chromosome 15, there is a cluster of genes that are expressed differently with what parent they were inherited from. So even though the genes are very close together, only some of the genes are turned on in chromosomes that were inherited from the mother, and other genes are turned on in the chromosomes that were inherited from the father. And that means that if you have a mutation in, for example, your maternal chromosome, the genes that are disrupted by that mutation are not going to be compensated for by expression of the paternal chromosome, because those genes are silenced on the paternal chromosome. And the opposite is true for Prader-Willi syndrome. So mutations on the paternal chromosome are not going to be compensated even though you have a fully functional copy of the gene on the maternal chromosome. It's completely silenced. So the way that the approach that these scientists were using to try to treat this is using a gene editor. So we talked about this very briefly before, CRISPR-Cas9 enzyme here. This enzyme is very good at finding a specific sequence of DNA and creating a cut in that DNA. In order to use it in this particular purpose, they actually deactivated it so that it could find a sequence of DNA, but it was no longer able to cut that DNA. And so that's why it's called dCas9, because it's deactivated. They use this to be able to hone another protein, this TET1 protein, to the sequence near the gene that they're trying to reactivate. This gene is silenced due to chemical modifications called methylation that occurs upstream of the gene, and this is telling the cells do not express this gene. And so their approach is to bring this enzyme that's tethered to TET1 over to this sequence. And what TET1 is going to do is it's going to remove those methylation marks. And now this gene is going to be available to be expressed. And when they tried this approach, you can see here, this green bar is cells that do not have a mutation on them. So they're expressing normal levels of the gene that's silenced in Prader-Willi syndrome. This is SNRPN. Whereas animals that have the mutation, cells that have the mutation, are in the blue bar here, and you don't even see the blue bar because the gene is silenced, and they're not expressing any of this SNRPN. But if the cells are given this dCas9 with TET1, they now are expressing that protein or that gene to about 10% level. And this 10% is great, but the scientists wanted to do better. So they reformulated, they tested other approaches. So here they tested another protein similar to TET1 called SUNTAG. They didn't see as much benefit there. They reformulated their TET1. They're on version 4 here, and now they're getting 50% expression. So the opposite of enhancing expression, you can think, well, maybe you want to silence the gene expression, which is good if your mutation is causing a gain of function toxicity. And so you can kind of approach it in a similar way to the enhancing expression version, where you can target the mRNA. In this case, instead of targeting the mRNA to stabilize it, you can target it to try to push it to degrade so that it's not creating the protein, and so it's not existing in the cell. You could also target the gene, and now instead of targeting the gene to try to push expression, you would target the gene to repress transcription. So one of the scientists in our lab was working on this recently, and she's published multiple papers on this. So Megan Kaiser, who now has just started her own lab, used this approach to develop a treatment for SCA1. And so in this approach, she wanted to deliver microRNAs. So microRNAs are very small RNA sequences that will bind to larger mRNA. So for example, when you transcribe Ataxin 1, you have an Ataxin 1 mRNA. And if you bind that to a microRNA, it can then push that mRNA for degradation. So in order to deliver this microRNA, Megan packaged it into an AAV. These two ITR bars here are those hairpins that I showed you earlier. And so here is the microRNA and the instructions for how to express the microRNA with this MU6 promoter. She also had to include other sequence just so that this gets packaged into the AAVs appropriately. And when she delivered this into mice that had human Ataxin 1 inserted into their genome, she saw that at varying increasing doses of microRNA, she got lower and lower levels of human Ataxin 1. So this was creating a decrease in the amount of mRNA that was available to produce Ataxin 1. One of the hallmarks of this disorder is neurodegeneration, which can be seen in humans, and it can also be seen in these mutant animals that Megan was using. And you can see in these animals, wild-type animals that don't have the mutation, have this certain width between in the cerebellum lobules. But in the animals that have the mutation, that width gets thinner and thinner and thinner. And that's a sign that the brain is degenerating. What Megan found was following her injection of her AAV with her microRNA, she was able to reverse this phenotype. She also performed a behavioral assay to see if these mice retained their motor function using a rotor rod. So what is a rotor rod? A rotor rod is this apparatus that we put our mice on, and it's a good way to test how well their motor function is working. So this dark gray rod here is continually spinning, and the mice are trying to stay on because they can't see the ground. Mice that have better coordination stay on this rod longer, and mice that have worse coordination fall, and when they fall, we take that time. And so you can see here, the higher up on this graph, the better the mice are doing on rotor rod. This black line here are mice that don't have the mutation, and all of these colored lines here are mice that do have the Ataxin 1 mutation, and you can see they're performing worse than the mice that don't. At 20 weeks following her treatment of the animals, you can see that particularly a certain set of the mice that were treated are doing much better on rotarod than all of the others. And these are this blue and green set here that were injected with 2 × 10 to the 9th vector genomes, or 8 × 10 to the 9th vector genomes. And so this is kind of the mid dose that Megan used. The higher and the lower dose didn't seem to work as well as this one. So, you know, in this case, what she showed was that there is definitely a window of dosage that would be most beneficial. Another way that you can lower gene expression is using ASOs. So ASOs are also small RNA, or I'm sorry, ASOs are small DNA fragments, and they bind to RNA, and similar to the microRNAs from the previous slide, they will then cause the mRNA to undergo degradation. In this case, you can see that this group was using mice that had the human Ataxin 2 gene installed in their genome. These mice also have a normal mouse Ataxin 2 gene that was present. And mouse genes are always, the first letter is uppercase, all the rest are lowercase, whereas human genes, all of the letters are uppercase. And here you can see that the mice that were treated with the ASO had complete ablation of their Ataxin 2 protein, whereas the mice, whereas their Ataxin 2 protein from their mouse gene has very low levels of expression here. So the ASO is targeting both the human and the mouse gene here, it looks like, but much more efficiently is targeting the human gene. And of course, when the mice are injected with just saline, they're able to produce both the human and the mouse version of the protein. The group also looked at neuronal function with these mice. So looking at the electrical activity that the neurons had as they're performing their normal functions, you can see that mice that don't have any mutation between 500 milliseconds are firing at a really rapid rate. So they have multiple spikes here. Whereas mice that have the Ataxin 2 mutation have fewer spikes in that same time window. The mutant mice that were treated with the ASO7, however, then reverse this phenotype and look more like the wild type mice. And they also performed a rotarod, and you can see the blue line here is the important one. So this is the mice that were treated but had the mutation, and they're outperforming the mice that had the mutation and were left untreated. I also want to point out here that ASOs are being used in multiple clinical trials, and in fact, 2 of them have been approved, one for spinal muscle atrophy and the other for Duchenne's muscular dystrophy. So another method you can think about that can help with certain disorders is introducing a new gene, one that is going to express a novel protein that may help enhance the health of the cell. So this group wanted to use a brain-derived neurotrophic factor, or BDNF, because they thought supplementing this in mice that have frataxin deficiency, so the underlying cause of Friedreich's ataxia, would be beneficial. And the interesting thing here, so they, I guess, did not have a mouse that had the mutation. And so what they did was they wanted to both decrease frataxin in order to model what this disorder looks like while supplementing in BDNF. So they actually injected 2 different vectors, one that would knock down the gene, and then the second that would introduce BDNF to be expressed. And BDNF is normally expressed in the brain, so this is just adding to that expression. And they did find that they were able to increase BDNF levels following this injection. And the other thing that they found was that these animals that were treated with BDNF but had lower levels of frataxin had processed these 2 proteins, caspase-3 and PARP1, in ways similar to animals that didn't have frataxin deficiency. But animals that were injected with just this pink vector here, so they only had frataxin deficiency and they weren't given the supplement of BDNF, they are cleaving these 2 proteins. So they're disrupting these 2 proteins far more than any of the other groups are. So this is showing that they're rescuing this particular molecular phenotype. And they also showed on rotarod that animals that had frataxin deficiency performed worse on the rotarod, but if they were supplemented with BDNF, they tended to perform similar to the mice that were just wild type mice. So the final strategy that I want to talk about is gene editing. So gene editing is a way that you can actually change the sequence of the DNA that's in the cell. And you do this with the various enzymes. One of the common ones is Cas9, which I've mentioned multiple times now already in the talk. So Cas9 is an enzyme that causes CRISPR editing, and CRISPR is clustered, regularly interspaced, short palindromic repeats. The name doesn't do it justice for what it actually does, but CRISPR is a method with which this enzyme, so this blue bubble denotes the Cas9 enzyme, it pairs with a small strand of RNA, this kind of purple sequence here. And that RNA will hone Cas9 to a very specific sequence, a sequence that is the exact complement of this 20 base pairs right here. And when it finds that sequence, it cuts at a specific site in the DNA. And so you can see these scissors here are cutting both the strand that is binding to the RNA here and also the other strand. And so these 2 cuts can be resolved in multiple ways. You can either give it a strand of DNA that you want to incorporate into that sequence, and it does that at albeit low efficiency, or following the cut, it will just kind of paste the two ends back together. And it can do that in a way that is a little error prone. So you can either delete a couple of base pairs or even add in a couple of base pairs. CRISPR has been retooled in multiple ways now also, to where people have deactivated it, like I mentioned in a previous slide. And so in this case, this Cas9 enzyme has one of the cutting sites deactivated, but the other site is still present. It also has a couple of new proteins added to it that will allow for this particular site in the sequence to be chemically changed so that you're actually changing a base pair from a G to a T or from a C to an A. And this is interesting because this allows for you to have a very precise edit of the DNA, and you get very minimal damage of potentially other edits occurring that you didn't intend to have. Cas9 has also been used to create even larger, very precise edits in a method called prime editing. So in prime editing, not only is a single strand of the DNA cut, like in the base editor, but now you give it another template in the RNA of sequence that you want it to incorporate at that cut site. And what it does is, using this other protein that was added to Cas9, it starts making DNA from this template of RNA. And that DNA then gets pushed into where that cut was. And now you have created an exact edit that you want to make at that particular site.
My apologies, Dr. Yrigollen. You have about 10 minutes left in your talk.
Perfect, thank you. So I am using a CRISPR-based approach in some of my research targeting the Fragile X gene, FMR1. And in this gene, we're targeting specifically the pre-mutation in order to treat the neurodegenerative disorder FXTAS, or Fragile X-associated Tremor Ataxia Syndrome. So to target this CGG repeat, what my strategy is to target both sides of the CGG repeat with Cas9 and two different sequences. So a sequence upstream and downstream of the CGG. So now I'm creating two cuts on either side of the CGG. And the hope is that this gets repaired by excising out those CGG repeats. And now you have a fully functional FMR1 without those repeats that are giving that toxic gain of function. In order to do this, I am delivering both the Cas9 enzyme and the two sequences to target Cas9 around the CGG repeat. To get over that size limitations of AAVs, I've packaged them into two different AAVs. So I'm going to deliver both of these at the same time to the animals. I've injected these into mice, and I see both molecular rescue as well as motor function rescue. So here is the rotarod assay that I did with these mice. And you see the black line here are animals that don't have that mutation. The red line here are animals that do have the mutation and were not treated. And the blue line are animals that have the mutation and were given this CRISPR treatment. And you see that this blue line is performing similar to the wild type animals, which we're excited about. Another approach that uses CRISPR for SCA3 gene therapy had a similar approach to me in that they were excising a large sequence around the CAG repeat. But in this case, they wanted to excise more than just the repetitive sequence and also the entire exon that repeat was in. And you can see here that they use that first approach of repair where they're giving it another sequence that they want to incorporate in here. So this sequence only has 13 CAGs and would not be pathogenic. And the result of this, when they tested it, was that they're able to see in in cells that they challenged by depriving them of nutrients. And when that happens in mice in cells that have this mutation, those cells exhibit markers showing that they're undergoing death. But when they provided this CRISPR therapy, fewer of those cells showed those markers. So they were able to rescue this this nutrient deficiency phenotype. An important consideration with any of these CRISPR therapies is the efficiency of your intended edit. So the intended edit here, they're trying to put this 13 CAG repeats in, and you see that it's actually quite low. So all of their intended edits here are under 10%. So if you think about it, 90% of the cells that receive this edit or this CRISPR therapy did not get the edit that would create this beneficial readout. And then for the final example that I'll give you, CRISPR-Cas9 can be used as another method of suppressing gene expression. And the way that this is done is by kind of creating DNA damage around the site of the gene that would cause the cell to recognize that this gene is damaged. And cells, when they recognize that a gene is damaged and it's creating a damaged mRNA or protein product, they will mark those for degradation. And so by creating many breaks along the DNA sequence around this Ataxin-1 gene, they're able to basically mark the mRNA and mark the protein for degradation. So this is called nonsense-mediated decay when mRNA is marked for degradation because of a damaged sequence. And at the protein level, the protein will undergo ubiquitination, which is basically marking it for degradation through a very important pathway of the proteasome. So you can see here that normally these mutations would not, so this gene has this mutation in it, and this particular mutation is causing a toxic gain of function, but it's not being recognized by the cell as being pathogenic. And so it's able to continue to create these dysfunctional proteins. But by creating these additional mutations, either a stop codon or a frame shift, they're able to evoke the nonsense-mediated decay or the protein degradation. And so with that, I am done with my examples. I've given you a lot of examples here, but certainly every single one of those gene therapy, gene editing strategies can be their own talk. So, you know, there's a lot of information here, but I hope some of this was helpful. Thank you.
Thank you for a fantastic overview, Dr. Yrigollen. This has been a lot of information in a short period of time. So we'll open the floor for questions now. Thank you to everyone who've already submitted their questions in advance. If you want to close your slides, that way people can see both of us at the same time.
Sure.
While you're doing that, a number of people have asked questions that I can cover very briefly about updates on specific trials, or if there's gene therapy in development for specific forms of ataxia. From a big picture perspective.
Oh, there we go. Sorry.
Oh,
Go ahead.
there you go. No worries at all. From a big picture perspective, the best way to find out information about clinical trials for gene therapy is to connect with your local patient advocacy organization in the US and Canada. That's the National Ataxia Foundation or Ataxia Canada, Ataxia UK, and Neuroataxia in Europe, or FARA for Friedreich's ataxia focused clinical trials. You can find the latest and greatest news there. There are clinical trials right now for gene therapy in Friedreich's ataxia and some forms of dominant ataxia, but it's still early stages. There's no approved gene therapies for ataxia at the moment, but getting involved in research is one option. And with that, we'll go to the more sciencey questions for you, Dr. Yrigollen. The first one is from an anonymous attendee. Why is it difficult to target the brain with gene therapy? After all, the genetic code is everywhere in the body. So would you have to deliver the therapy to the brain specifically?
Yeah, so there are a couple of reasons that it's challenging. One is the brain is very well protected by a structure called the blood-brain barrier. So just injecting gene therapies, for example, into the blood with like IV administration, often will not get to the brain because the brain will, the blood-brain barrier will act like a wall and it will not let it pass through there. So some of the AAVs now are getting good at crossing the blood-brain barrier. But another strategy is to kind of circumvent that by directly injecting into the brain and into the cerebral spinal fluid. And so if you do that, you can get more access to the brain. But even with that, the brain is a challenging organ to deliver gene therapies to because there are so many structures in the brain and so many different cell types. And actually, some of those cell types you don't want to touch, like they're very sensitive to any kind of intervention. So there are a lot of different challenges with trying to deliver to the brain.
Thanks for clarifying that. We've had a few people ask a very similar question of in a hypothetical situation, if they receive gene therapy to treat their form of ataxia, would that correction be passed on to their children, or would their children would also have to receive gene therapy? From what we
Yeah,
know in mice, could you speak a little bit about this?
sure. Good question. I'll tell you that the FDA is pretty adamant, and I shouldn't speak for the FDA. I am not an FDA employee, but I will tell you my experience is the FDA is pretty adamant that none of these gene therapies should transfer to germ cells, and that is to say, should be able to be inherited by your children. So any of the gene therapies that they would approve, I think they would make sure that they would not be able to be transferred to children. So, basically, they don't want to have a gene therapy potentially cause a problem and then that get transferred into children. So, you'd have to give the gene therapy, and then if your child got the disorder, they would have to get the gene therapy as well. But that's a good question.
Thanks for clarifying that. We have a few people asking this
And
question around.
I said,
Oh,
I'm sorry, I said the FDA. I did hear all of the people from all the different countries. I think it's same for most of the big regulatory bodies, but
mhm.
obviously, per country that might be different. But I think that that's also kind of the bioethic consideration right now is that we don't change the germ cells.
That makes total sense. A few people have asked a similar question around when these therapies are approved, when would be the best time to receive treatment? Would gene therapy be something that people receive before they start having symptoms? Could it reverse symptoms? From what you know in the mice you're studying, could you speak a little bit about this?
Yeah, that's a great question. So, generally speaking, I think that when you're talking about neurodegenerative diseases, and therapies that can be kind of invasive and expensive, or if you're talking about diseases that don't have complete penetrance, so not everybody who has the mutation is going to get the disease, you probably don't want to say earlier is better because it's possible that you'll never need to get the therapy. That being said, in a lot of diseases, earlier is better because you're able to correct the problem before cells that you aren't going to be able to replace start dying off. So, neurons, if you start losing neurons, you're not going to be able to replace those. And so you'll maybe be able to reverse some of the symptoms, but maybe not all of them. So I would say, it would probably be case dependent, certainly, but I would say for neurodegenerative disorders, you would probably want to do it early in symptom onset, or if people knew more about the trajectory of symptom progression, you would maybe know, is there a window where I should really get it versus maybe wait a little bit.
Good to know. So
And
there's
I'm
over
no,
300 types of ataxias. You need to
yeah,
take into account the differences of all these different kinds of disorders.
and I'm no medical doctor, so that's my own opinion, not medical advice.
No, that's good to know. Please talk with your own MD before doing anything if
Yes.
and when these medications are eventually approved. We have time for a few more questions. We have someone who's asking, Can gene therapy be used to treat ataxias of non-genetic origin, such as acquired ataxia? Is there anything known about that?
I think that they probably can. So it would be, you would take those different tools and maybe you definitely have to understand the biology of the acquired ataxia to be able to use a gene therapy. So you're going to want to know, well, what proteins are being dysregulated that we could potentially add in with a gene therapy, or what proteins should we target to get rid of because they seem to be causing these problems. But I do think that other forms of ataxia definitely can benefit from these same strategies. It's just that you have to be really smart about what strategies you're going to be using.
So it sounds like more research is needed, but very good to know. We have a question. Could you please explain more about what you said on the low percentage of editing on one of the slides about CRISPR therapy? This attendee wasn't sure what it meant and if it's efficient versus effective or something else.
Yeah, that's a good question. So my highlight of that is CRISPR is great because you're able to get to the DNA. You're able to actually change the thing at the root cause that is broken. But the problem is whether or not you're able to change it in the way you want to in a high enough number of cells. So a low editing efficiency might be okay if you need very few cells to produce the protein to have a robust effect. There are disorders where you only need like 6% or 10% of cells to be fully functional, and you actually get a really pronounced improvement in symptoms. But there are other disorders where you're going to need that amount to be much higher. And, depending on the disorder and what's known about it, people might know exactly how much correction you need and be targeting that level of efficiency with their gene therapy.
And then I realize we're slightly over time, so thank you for bearing with us. We have one last question. We have a couple of attendees who've asked about the invasiveness of these kinds of injections. As right now, most of what you discussed were in mice. When this is scaled up to human beings, what would this kinds of treatment look like in terms of how frequently would people have to get injections? What would the
Right.
injections be like? Could you speak a little bit about that?
Yeah, so it depends on the strategy. So any of the like LNPs, ASO work, those are molecules that will go away over time. So if the change that they make is not sustained in the cell, so if the cell kind of maybe loses whatever advantage was created, then it might need to be redosed. The idea with any of the gene editing strategies or strategies that are using AAVs is that those are going to persist over time. And so we like to think of it as kind of a one and done therapy.
Well, it was very interesting to learn about the variety of gene therapy options that are currently in development. So thank you again, Dr. Yrigollen, for sharing your expertise and your time with us today. We really appreciate it.
Thank you so much. And I hope that other people found gene editing and genetics as exciting as I do.
And thank you for attending this session today. I hope you found something interesting or helpful, or maybe even both. So thank you for spending this past hour and a little bit with us. We hope you have a wonderful rest of your day. And wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful day ahead. Take care, everyone.